All right, I think we left off around here. We're looking at cell wall structure, and what do you think? Gram positive or gram negative? For once, it's not my laptop doing that kind of stuff. Gram positive or gram negative? Positive. Lots and lots of layers of peptidoglycan. Okay, so chainmail like configuration. What are the sugar derivatives? They're 3-letter acronyms. What are those? NAG and NAM. That's how I say it. So is it neg? And. What is it?
Alternate.
Yeah, alternate. Neg. So if I ask you to describe that, you have to say that. Just don't say, oh, they're neg. I say, well, it won't be on this exam, but on the next exam. If I ask you, just don't say, oh, neg. You have to be like detailed, like, okay, yeah, but, okay, so neg, long chains, right? And then off of the NAM sugar, what's dangling off of that? In general, just there's, so you have the neg, that's hard to say fast, neg, and then off of each of the NAMs, there's something that's coming off of it. Amino acids, right. So there's 4 amino acids coming off of one of the NAMs, and then you have to imagine another chain of neg and NAM, and then off of that chain, half of its NAMs are 4 amino acids. And so you have them cross-linking. That's how they can stay cohesive and not kind of fall apart from each other, is you have those peptide cross-links that allow for, let's go to this one, that allow for the neighboring chains to hook to each other. Right, so neg, and off of each of the NAMs, purple ones, you see the 4 amino acids of the one bonding with the 4 amino acids of the neighbor. Okay. And then it kind of looks like a chainmail configuration. And we saw with that first picture we looked at that the gram positive is pretty thick. So I know in lab yesterday you did not do the gram stain. I'm certainly hoping we got that ready to go for tomorrow. And so when you run that test, that staining procedure, you're keying in on differences in the amount of peptidoglycan. And that's one of the main reasons why we call it a differential stain. You'll be able to use 2 different stains. And because of the difference in thicknesses, one type of the gram positives will stain purple and the gram negatives will stain red. So you'll learn about that. And so we talked a little bit about the gram negative. I think we left off pretty much around here, where we're looking at the idea of the gram negative cell wall. And it actually, even though we say it's a cell wall, or some people say it's a cell envelope, it does have this funky outer membrane as part of its cell envelope or cell wall structure. So this is where we left off, looking at the overall structure. And so way down on the bottom, that's your cell membrane. And then we have to think about the space above it. So that's the periplasm. That's just the space. And right, they drew the peptidoglycan in there, which is true, but there's all kinds of proteins and enzymes in that space too. It's a very busy area. It's like a superhighway, very busy. And then you see this outer membrane. And you can definitely tell that outer membrane doesn't look anything, well, doesn't look like the cell membrane. Although if you look here at this side of it, that kind of looks like a lot of what we see going on in the cell membrane, but this outer portion of the outer membrane, very, very different. So that's the LPS, lipopolysaccharide, and you can see that there's different colors of these hexagons. Those are different sugars. So I want to work through, I want to start on the outermost portion, which is this portion, the outer leaflet of the outer membrane. Okay, that's what we're going to zoom in on next, which is Okay, so the outer leaf of the outer membrane, that's what we refer to as the LPS portion, the lipopolysaccharide. And if you work your way from the very outside and work your way down a little bit, some of those sugars at the very outside, we have a few of those, and those are called O-specific polysaccharides. And I'm not gonna, this information here is just in case you're curious, I'm not gonna ask you that. But you do need to know, yes, there's O-specific polysaccharides on the very, very outer portion. And then those are bonded to the core polysaccharide. Again, you don't need to know the details, but if you're curious, that's the chemical. And then it depends on the species, they kind of mix it up. And then the lipid A, that's the portion of the LPS that our body can respond to pretty negatively. It can be toxic in high doses to us. It can set you into shock. What's that kind of shock where you just go into super shock? There's a term for that. I'm blanking. Is it septic? No, I don't think that's the one. Is it? You might be right. I'm blanking. Obviously, it's not that important. But anyways, so the lipid A portion, I'll show you a picture of it too. We'll go back to the drawing. They've determined that that's the portion that our immune system responds to, and it can set our immune system to like overreact, almost like an allergic reaction. And it can be problematic. It's actually categorized as an endotoxin, and we'll get into that. You've already heard a little bit about these endotoxins. We'll talk more about those when we get into microbe-human interactions. And then there's fatty acids attached to the lipid A, just typical fatty acids. Okay, so O-specific, core-specific polysaccharides, which are then bonded to the lipid A, which is then bonded to the fatty acids. So the drawing here is trying to show that. So the pinkish ones or purpley ones are O. I guess those are purple. And then these are more pink. The core are pink. And then you have the lipid A with the fatty acid tails up with that. Okay, so that's composing the outer membrane, the outer leaf of the outer membrane. So what we just described is right in this. Well, okay. So the lipid A are the bright yellow squares. That's what our body reacts to. So if it's an intact cell wall, if the gram-negative bacteria is alive and it's all intact, we don't really get exposed to the lipid A. So we can, like right now, you have trillions of E. coli that are gram-negative in your intestinal tract, and you're not in any danger. But if all trillion of them, or maybe a subset of that, died all at once, and then all that lipid A gets released as the cells break open, then you might be in trouble. So it's only really when the lipid A is free and floating around that you get into some danger. Okay, then let's describe the next part, which is the like bottom half here. So this region. Okay. So if you look at that portion, you see a lot of what looks like phospholipid fatty acids. So that's something familiar. So that's our inner leaf area, the inner leaf of the outer membrane. So it has the phospholipids, like how they drew it. And then there's these structures called lipoproteins, and that's basically a way to anchor the outer membrane to the peptidoglycan, so it's stable and doesn't like fall apart. So the lipoprotein it's lipid and protein interactions going on there, so that you can have it hook into the peptidoglycan. And then I'll point out again, we'll go back to the other picture. There's these porin proteins. that they've discovered a while ago. And they're transmembrane, which means they span the entire width of that outer membrane. And they're funky. They add a different kind of three-dimensional conformation and their subunits. And they have discovered that there's different kinds that are adapted to allow certain molecules to come in, but not others. So this is one way that the outer membrane has some selectivity on what's going on with these porin proteins. And so, like I said, there's different kinds. They give them different acronyms. And basically they allow for small molecule transport back and forth, in and out of the cell. And we go back to that picture, you can see they tend to have this conformation where there's these three major subunits that cluster together, the channel proteins, kind of three big tunnels. That's how they actually look when you look at them under the electron microscope. And so those are the proteins that allow for transport. And you have your phospholipids here, and then lipoproteins are anchoring into the peptidoglycan. All right, so that inner leaf is relatively similar to what we see in the cell membrane. Not identical, but. All right. Then let's look next at the space. What's going on in the periplasm? So the periplasm is just the physical space between the cell membrane and the outer membrane. And it's not very wide, but it's decent, 12 to 15 nanometers wide. It's considered to be gel-like. It's not like super watery because it's chock full of stuff. It's got a lot of proteins and enzymes. And so it does give this kind of thick, more viscous when they run experiments on it. So there's lots of different enzymes and proteins. Hydrolytic means that they're breaking things down as they come in into smaller bits and pieces. There's binding proteins that will be binding with chemicals coming in to then typically kick in a signal cascade, but there's chemoreceptors. There's a lot of stuff in there. And then part of, if you look in the periplasm, there's the peptidoglycan, but it's pretty thin. Okay, so when you say the cell wall of gram-negative bacteria, you're taking in, consider the periplasm as part of it. You got the periplasm, and then you have the peptidoglycan in there, and then you have that outer membrane. Questions on the gram-negative type? Pretty funky. So chapter 3 goes into more detail than what I do on especially the cell wall. So really zoom in on what I'm covering. All right, because it's it's good information, but it's not, I won't go into that level of detail. They put in a lot more stuff in there. So just be aware. By the way, under the electron microscope, here's what a gram-negative looks like. So you can just barely detect the peptidoglycan there in the periplasm area. So very different. Okay, so I'm not going to get too much into the acid-fast structure in the lecture. You'll get a little bit of that in the lab because we'll do an acid-fast stain. Probably tomorrow if the right bacteria grew up for that. And so tomorrow in lab, you're going to be hopefully doing the gram stain and the acid-fast stain. Okay. Now, archaea. Let's think a little bit about them. They don't have as much research going into their compared to gram-positive, gram-negative, but it's catching up. So they've discovered that archaea cell walls vary pretty dramatically. It's not so consistent like what I've been saying about the gram-positive versus gram-negative. And that has a lot to do with just where they live. They have different adaptations they have to do in order to survive. And so some archaea cell types will have what we call pseudo peptidoglycan. What does pseudo mean in general? False, fake. Okay, so they're trying to say this is not true peptidoglycan. Like if you look up the word peptidoglycan, they'll formally define it. This doesn't fit it. Okay, so it's pseudo peptidoglycan. So archaea don't have peptidoglycan per se. They have pseudo peptidoglycan, and that's because when you look up the definition of peptidoglycan, it says it has to have NAG and NAM. And you don't see that in the archaea. They have NAG, but they have NAT instead of NAM. If you really, really want to know what NAT stands for, I don't even know if I can pronounce it. I have it written down. You're not expected to know it because it's ridiculously long. Where is it? N-acetyltolosamine acid. There you go. One and done. I'm not doing that one again. So NAG and NAT. It's pseudo, it's fake. It's not true peptidoglycan, but it's kind of similar in the sense that you have NAG, NAT, NAG, NAT, NAG, NAT, and you still have some same similarities with the overall three-dimensional structure of what you see in peptidoglycan, but it's NAG and NAT. So it's pseudo. The other thing that's different is they don't have the beta-1,4 linkage between NAG and NAT. It's beta-1,3 because of how the sugar derivatives are chemically made. It has to be a beta-1,3 linkage between them. And that means they're not sensitive to our lysozyme. And so you can eat archaea and they'll make it through your mouth. Your lysozyme enzyme won't chew up the peptidoglycan, so to speak, or degrade it. So here, if you're curious, this is what the beta-1,3 looks like. We have the NAG and NAT. NAG, NAT, beta-1,3, which lysozyme can't touch, break down, but you can see you still have this peptide crosslink idea. Okay. Now, you know, having said that our lysozymes don't attack the beta-1,3 bond, that's okay though. Because if you happen to eat an archaea, like if you were a little baby and you ate some soil as you're crawling around, no big deal. It's actually good for babies to eat a little soil, actually. But archaea aren't adapted to you, so they would just not make it. They're not part of our internal or external ecosystem. All right. Then they've also discovered some archaea cell types have just a sugary polysaccharide layer that covers their cell membrane. Some can have glycoproteins as what they consider the cell wall. Or maybe it's all protein. And then the picture that I'm pointing to here, that's a nice picture of what they call the S layer, which is paracrystalline surface layer. There are a handful of bacteria that can do this too, but way more archaea. And you can see that it has this nice hexagonal shape. So these are all protein subunits that are bonded together, and they call it the paracrystalline S layer. And that is actually, of all the archaea they've studied so far, you'd think maybe pseudopeptidoglycan would win, like more cells have that than anything else, but no. So far they're saying, it seems like the archaea really like to do the paracrystalline. That's their strategy, that's their go-to, unless they're forced into some other adaptation. So at least so far, that could change over time once we study more of the archaea cell type. Like I said, the research is a little bit lacking compared to the gram-positive and gram-negative type.
So some archaea have pseudopeptide, like some have polysaccharide.
Yes, so these are all options, different flavors of cell walls.
So it's not all three of polysaccharide glycoprotein.
Some can. They mix it up. They're really quite diverse in how they run their cell walls. So, other questions on the funky archaea? All right, I'm going to shift, yes. Yes, this overall structure does give them certain advantages, yeah. And so it, the pseudopeptidoglycan is pretty nice too. So really, probably the paracrystalline, the reason it's so popular amongst the archaea is that so many archaea that have been studied so far have been taken from what we call hostile environments, places we can't deal with. And so right now, this is winning because we like to go to the hostile places and see how are they doing that? And this is a good one. So. Okay, I'm going to shift into the next slide set because otherwise this slide set would get too big. So I'm going to go into part 2 of cell structure. Is that on EDUCAT? It should be, right? It's there, right? Okay, thank you. Okay, so basically I've kind of started from, the very basic of what is the cell membrane, kind of refreshing your memory on that, and then looking at the cell wall. But now I want to look at some other like appendages and things that some microbes have that can really help them adapt to their current habitat. So one of the most famous things is the flagella. And if you just talk about one as flagellum. So surprisingly complex in its components, and it doesn't work like eukaryotic flagella. So you've probably seen video of like protozoa that are eukaryotic under the microscope and they're swimming around, and it looks like, or like sperm cells that are swimming around, and it looks like the flagella is whipping around, right? And that's not how bacteria move their flagella. Their flagella is actually corkscrew or spiral shaped, and it's rigid, and then it just kind of rotates, kind of like a propeller on a motorboat. It just rotates, and that's what gets them to move from point A to point B. So if we start where it's anchored, we call their anchoring region the motor portion of the flagella. And that is right into the cell membrane. And then I'll show you a picture of this. You'll actually see a ring-like structure. And in the middle of the ring, in the whole area, then there's what they call the central rod. And that is surrounded by these so-called MOT, I say MOT, maybe some people say MOT, I say MOT, MOT proteins associated with the motor. All right, and then what they do is they allow for the rotation. They cause that torque force that allows the part that sticks out, which is called the filament.
What's the ring system?
I'll show you. So the ring system, it's literally a series of rings that provides a space for the rod to come through. I'll show you a picture. It'll make sense when I show you the picture. And then we're going to also see that there's these special flea proteins, they're called. There are a bunch of flea proteins that are right there by the motor. And I'll point those out. And so they say they function as a motor switch. What that means is the switch is, do I want to rotate my flagella clockwise? Or do I want to do it counterclockwise? And if they decide to change, what I'm going clockwise, now I want to do counter. They'll do the motor, the flea proteins will do the motor switch on that. And then there's going to be that, you're going to see a hook region. This will all make sense once, but I just wanted it down in words first, and then we're going to look at the picture. And then the actual part that sticks out of the cell that you might see coming out is called the filament. So taken all together, this is the flagella. But you can see there's lots of components that make that up. lots of proteins. Okay, so this is a heavy investment in energy and resources for the bacteria to do this. When you look at the filament and really study it closely, it's just a bunch of subunits of protein that they've given the name flagellin. At least they named it something that makes sense. So the protein is called flagellin. A bunch of flagellin subunits build that entire filament. Okay, that's a lot. So let's actually look at a picture of it. So here we go. So here we have okay, so you have the MOT protein or the brown sausage looking things. And so you have to imagine they've kind of cut it away so you can see the other parts, but those go all the way around here. In the back there, so they just go all the way around. And then you can see that there are these different rings and they named them, you know, C ring, MS ring. You don't need to know that. Just know there's rings. Can you tell what kind of cell type they're drawing here? Gram negative, okay, because you can see that there's a really thin peptidoglycan in this periplasm area. Okay, so here's your MOT proteins. Here are your rings. With the gram negatives, they tend to have more rings than gram positives, but you don't need to know that. Just know there's these ring structures, and then the rod actually goes through that. Okay. So the yellow portion there, sticking straight through that, those are the rod areas, the rod structure that's coming through the rings. Then that rod needs to connect smoothly to help and to like maximize rotation and the force and everything. So they have a special hook region that's kind of curved. And that's just to help with getting the filament and getting it, you know, the torque force to work correctly. So it's reminds me of that. You remember watching that cartoon on, you know, the ankle bones connected to the whatever bones. You can tell I haven't taken animal or human. You know that song? Okay. Bones connected to this bone, which is connected to that bone. That's what I always think about. Although clearly my memory is bad on that right now. But anyway, so your rod connected to the hook, connected to the filament, and all those brown little balls there, that's all the flagellin subunit proteins, all building that filament. So this is a lot. Now you need to have a torque force. So that purple arrow here is trying to show that especially where the hook area is, that really does allow for some nice, you know, kind of beginning of the action of the torque force. Okay, did we cover everything? Let me look back. Now, where is that? Flea. Oh, yeah, the small ones. Yeah, the flea proteins. These are the motor systems. So the MOT proteins are kind of round, and then inside of that are the flea proteins. And then you have your rings, your rods, your hook, your filament. So the MOT protein is, they must make that short for motor, I'm guessing, right? It's so maybe you should call it MOT, but I can't because one of my brother's name is Tom, which is MOT backwards. And my other brother used to tease him all the time. And so he got the nickname MOT. My brother Tom got the nickname MOT, and I can't get that out of my head. So that's, anyways, it helped me remember this whole thing. But anyway, so maybe it's MOT proteins, which are part of the motor idea. Okay, and the flea proteins there are the motor switch, which change the direction. I think this is from the current edition of the textbook, which is also correct. It's just a slightly different drawing, but it's got pretty much everything there, a little bit updated. But the other thing I wanted to point out here is figure B. So figure B is looking at, they got this purple thing going again, which is trying to show that, okay, we have to have some way of creating a torque force. So that's not going to come for free. Okay, so what do you see happening? What's flowing through the MOT or moat proteins? Protons. Okay. They're trying to show a proton gradient. The artist should have been a little bit more flamboyant and put lots of protons on this side compared to the other side. So you have protons flowing through the MOT or moat proteins. So it's a proton gradient. That's like potential energy. Once you establish a proton gradient, that's like potential energy that can be used to do something. We talked about it being used to allow for ATP synthesis. This is another way it can be used. So proton gradients across the cell membrane, cell wall of the bacteria can allow for flagellar movement. So that's what they're trying to show here. That plus minus is also just trying to show you need to create this proton gradient, this electrochemical proton gradient. And as the protons flow through, that generates the torque force and allows it to rotate. Now, the study that I read, and maybe it's in your book too, they're claiming that you have to have 1,000 protons pass through to get one rotation. That's impressive. That's a lot. Yeah, I'd like to find another source to verify that because that just seems like so much. But that's what they're claiming. And then another paper I read, so far anyways, they know there's 40 different genes that have to be turned on just to build this structure. So it's a lot of effort. So now you can start to appreciate back the other week when I said some bacteria decide to not build it if they don't need it. I get it now, this is a lot. So if you don't need it, you might want to hold off and just put your resources elsewhere. All right, so let's assume it was built correctly. The torque force is happening. How are they moving? Well, turns out it depends on how many flagella the bacteria have and where they're built on the cell of the bacteria. So here you can see, let's start with the one on the bottom, polar. Polar, if you say, oh, look at that cell, it has a polar flagella. What you mean is just one flagella, and it's usually a rod-shaped bacteria, and the flagella is just at one end. Okay, then you say it's polar. If you look at them under the microscope, a bacteria that has polar flagella, if you look at them under the microscope, they look like they've had a whole pot of coffee. They'll be zipping along and then they'll stop for a second and then zip in a different direction, but they go really fast and jerky. And that's what they're trying to show here, that it rotates, it goes in one direction and then it stops, and then it'll just stop and hang there like Brownian motion will kind of cause it to shift direction, and then it goes again. And so you'll see these herky-jerky quick movements. And then if you look at more of the paratrichus idea or a Lophotrichus, which I'll show you in a sec. Paratrichus, that type, when they don't really, well, okay, I'll show you a better picture of paratrichus. Here you're seeing several flagella, and you can see they're trying to show that it's weird, even though they have many flagella, they coordinate the flagella kind of together, kind of bundle them together, and then coordinate the torque direction and amount so that it's very coordinated. And here you can see that they'll start to move. And usually if they're paratrichus, if they have more than one flagella in general, when you look at them under the microscope, they tend to be more slow and stately, like a big cruise ship. They're going nice and slow, and then they'll stop and their flagella will kind of come apart from each other. And then they'll rotate a little bit, and then their flagella will come back in unison and they'll head off again. but all of that is much slower. So just by looking at them under the microscope, you can at least get a feel for, do they have one flagella as they're moving, or do they have more than that? And that gives you a little bit of idea. And then here you see this reversible flagella in the middle here where they're going, first it's going counterclockwise, then it decides to go clockwise, and that actually changes the direction by that movement. So let's look at a little bit different view here. So polar is in the middle, just one coming off the end. And then Lophotrichus is several coming off the end. Okay, several of them, you'll need to know that term too. And then Paratrichus, that's a pretty good image. Paratrichus is, it's just so bizarre. It's all these flagella coming off all the way around the rod-shaped cell. I don't know why they adapted that strategy. It just seems flamboyant and not terribly efficient, but apparently it works. All right. So there's different types and amounts in terms of different types of where they're located, different amounts of flagella. Here's some nice pictures there too. I mean, look at that. How many flagella? That's crazy. It's just over the top, but some do have that. E. coli has that. Pardon? Well, you would think they would, right? I mean, I can imagine where that would happen. They are rigid though, so it's not quite as easy to tangle them. That looks pretty tangled though. That is an amazing stain, by the way. Did you look at flagella under the microscope, the prepared slides? I used to try to have students, when I taught the lab myself, we try the flagella stain. Oh, it was just awful. It was, the success rate was like 1 student out of 24 would get it to happen. So that's why I ordered prepared slides, because it's very tricky. So, okay, so flagella, a pretty interesting invention.
They're rigid, they stay.
Yes. Yeah, they always stay helical. Is that what you're asking? Yeah. So there are other ways to move. So the next one we'll get into is gliding, but I think I'm going to stop here because I wanted to leave a little bit of time in case you had questions regarding the exam. And so I have a pretty solid draft right now, and I think I'm still going to add a couple more questions. Right now it's sitting at around 70 points total with 1 essay, but I'm going to add 1 more essay. I just haven't decided exactly how to word it and how much points it'll be. So the final exam point value is somewhere between 70 and 80, I would imagine. And with probably 2 essay questions, 1 longer, 1 shorter, I'm guessing. And then roughly 30, I think, multiple choice, roughly. So it's designed to be done within 50 minutes, and it shouldn't be a problem. However, we are, this room is open after we're done on Fridays. So I can stay a little bit later if I have to. Oh, and Duncan will be here too. So between the 2 of us. So basically when you come in, please come in quietly. I'll try to get here right at 10:02. And if the other class gets out of here quickly, you can start right away. So having said that, be aware people might already be starting when you walk in. And then most people do the multiple choice first, but you don't have to. But if you do that, you pull up the multiple choice and start going through it, and then you submit it, then close all the way out of your browser and then go back in. And that way it will kind of clear out the respondus issue and you'll be able to get back in no problem. If you keep it all open and try to hit on the essay portion, it'll glitch on you 9 times out of 10. So just close all the way out and come back in, then click on the essay portion.
You want to not submit it, but just.
No, submit it.
Just close.
No, submit it. Submit it and then close all the way out. Close your browser. And then before you leave the classroom, you need to show either me or Duncan that you submitted both versions of the exam, the multiple choice and the essay. So make sure that you do that before you leave. If not, we'll flag you down. Okay, and then remember, it only goes through that one slide on peptidoglycan, though, just the big picture idea. So does anyone have questions on actual content material? We started way back with the microbial evolution slide set. Yes,
Just like a review on the beginnings of the metabolism.
yes. So let me pull up the microbial evolution. Okay, so okay, so the beginnings of metabolism, right? That's the one. So this is considered hypothesized to be one of the first biochemical reactions where they're putting together a series of steps in order to accomplish the goal, which in this case is to get some energy, some ATP. So in this case, they don't really care so much about how they do that. They're just, I like to think of microbes that have adapted to all these different environments. It's kind of like a chef who walks into a walk-in pantry and they're like, what are we having for dinner? What do we have available? And they just sense what they have around them and start working with it and create this beautiful meal, in this case, with the goal to be able to get ATP and dine on some ATP. So they have, in billions of years ago, they had ready access to iron and sulfur and hydrogen, no oxygen. That wasn't there. Methane was there though, but not oxygen. And so it's like, okay, we got to work with what we have. Let's see what we can do. So outside the cell, there was plenty of these materials available in their environment. And also have these reactions happening with ultraviolet light coming from the sun. But basically, when you look at these reactions, the key thing, one of the key things is the formation of hydrogen gas. And you keep that in mind as we work forward. Hydrogen gas is pretty cool and microbes really love it. A lot of microbes really, really like it. And so in this case, you have the microbe stripping electrons off of it. So then if you do that, what else are you left over with? Protons. Okay, if you strip electrons off of hydrogen, you're going to get protons. Well, right there, that should pique your interest because we know that our cells, including microbes, they really work with protons. That's a good thing. That's a good thing to have. And then here, if you see this reaction cooking along outside, so yes, you got the hydrogen gas being made, and then the electrons get stripped off of it and fed into this internal reaction going on. So again, the microbes really aren't like the hydrogen sulfide, they don't really do anything with that. So they're using these electrons, they're running this reaction, they get to hydrogen sulfide, that's toxic. They're like, yeah, I don't really want that. So they put that out of their cell, but that's really cool because it keeps that reaction moving forward. Yes.
Does the hydrogen gas go through the primitive hydrogenase that strip the electrons off?
Just the electrons. So the hydrogenase does like interact with the hydrogen and the electrons get, I say, stripped off and then they get fed into this reaction. But the protons stay out. They stay out.
Are we going to know that chemical reaction?
No, not from scratch.
Just like.
Like recognize it, like be able to recognize these, but you don't have to write it out from scratch, you know what I mean? So, again, the artist was a little not flamboyant enough with the hydrogens or the protons, I should say. Because if you keep this going over and over and over, where you strip electrons off, feed them in, get this reaction, get your hydrogen sulfide, that feeds into this reaction, and you just keep doing that. Round and round and round and round, you're going to get a big proton gradient. And that's like the goal. And then once you have that proton gradient, the only way that they can even out across the membrane through the laws of diffusion, they have to go through the ATPase, because they can't go through the big hydrophobic region. Charged molecules just can't do that. They have to go through something that can work with the charged proton and allow them to come through. So they go through that ATPase. And then once they do that, that actually creates a torque force as well. There's a torque force that occurs here that allows for ADP and the phosphate to actually physically interact. Questions on that? Questions on anything else with, yes?
I think where it says 95% or greater 60, that's on R and
Yep, so. Is that in this slide set or the next one? No, it's fine. So basically that is the 95% rule. So that's the rule where they say if you have sequence similarities, so the DNA sequences, when they go ahead and do that sequence comparison, if they are match up, so 95% are the exact same nucleotide sequence, then they say this must be the same species we're comparing. It's two different, organisms. It's 95% or greater similarity. It's the same. Maybe it's, E. coli. This is E. coli. Yep, this is E. coli. They're the same. Other questions on any of the content?
Do you have like major takeaways that you want to heard from Lenski's experiment?
Oh, Lenski's experiment. Yeah, that's a good one. So those are slides. I didn't personally cover it with you in here, but it is in the slide set, the first slide set. Second. Oh, there it is. So they did this really cool experiment. They're still doing it, I think. He must have a lot of workers in the lab, but basically growing E. coli. So you're going to learn a little bit later in lab that E. coli is famous. In fact, I think that's how they define E. coli. One of the ways they define it. It's famous for not being able to use citrate. Citrate is a sugar or an organic acid that is big. It's too big for E. coli to use. They don't have the enzyme to break it down. They don't use it. When Lenski was doing this experiment with the E. coli he had, what they did was they grow it up in broth and then they'd subculture it routinely into fresh broth, keep growing it, subculture, grow it up till it gets really cloudy and full of cells and subculture, keep doing that over and over and over and over. And then periodically take cells and freeze them and then start sequencing them. And he'd be looking for, are there mutations going on? Are there changes going on? And this data here is showing a big change. And one of the things that was huge was this ability to use citrate. So they were called citrate positive cells. That means they're able to use citrate. And to you, that may be like, well, big deal. It is a big deal because as long as we've studied E. coli doesn't use citrate. So this was a big deal. And then of course, with all the DNA sequencing, you can show how that occurred. So that's basically the big picture idea of what's going on there. So it's evolution in real time. I mean, within Lenski's life, he was able to see basically a new E. coli, one that is able to use. All right, so make sure to ask questions during the exam. I try to help as much as I can. And then today's Wednesday, so I'll be checking my email into the evening hours leading up to the exam. So if you do have a question as you're studying, you can send it to me and I'll try to get back to you as soon as I can. Okay. Plus office hours and stuff like that too. But if you're in the evening hours, I will be checking.